Hello students, this is Dr. Faizan Mirza. In this current video, I'll be discussing the second part of the cell structure and microscopy, in which I'll be discussing with you the ultrastructure of the animal cell and the organelles such as nucleus, the endoplasmic reticulums, and Golgi mainly. So let's start with what your electron microscope can show you of the ultrastructure of an animal cell. So when you look at the animal cell using an electron microscope, you know there's a high resolution, there is a high magnification as well. So we see nucleus, yes, but now nucleus will appear a bit different from what actually it appeared in a light microscope. You have a nucleolus, chromatin, nuclear pore, nuclear envelope. There's two membranes can be visible. You can see that both membranes of nuclear envelope can be seen in this structure right now. In light microscope, we were not unable to resolve this into two separate layers. Here you can see these two layers of the nuclear envelope. Apart from this, we have rough endoplasmic reticulum. This structure was not visible there. Rough endoplasmic reticulum is always joined to the nuclear envelope. This is where it grows from, and it will have these ribosomes attached on its surface. Ribosomes are present in the cell, in the cytoplasm, on the rough endoplasmic reticulum, as well as on the nuclear envelope. Where are the ribosomes produced? The ribosomes are produced inside the nucleolus. So this nucleolus is something that produces ribosome using its own DNA. Once the ribosomes have been produced, these ribosomes start to move out of the nuclear pore and they attach to the nuclear envelope on the outer surface, and then they become part of the rough endoplasmic reticulum where they can carry out protein synthesis. Together with rough endoplasmic reticulum, we have smooth endoplasmic reticulum. This is not having any kind of ribosome attached on its surface. This is also attached to the nucleus, although in this diagram, the connection between the nuclear envelope and smooth endoplasmic reticulum is not shown. Minded that this connection might not always be visible to the to the eye or to you because of the kind of section that was taken of the cell. So yes, it might be visible, it might not be visible in that diagram, but this is always the case. Nuclear envelope is the site where the rough ER and the smooth ER are joined to. So, and they basically arise from there as well. So we have mitochondria, and the inner detail of mitochondria can be seen. This is the mitochondria along its vertical axis. This is the mitochondria along its circular axis. So you are looking at it on a different plane. You are looking at it on a cross section, and this one is being looked at along its length. And as I mentioned, this one is being looked at along its cross section. We have lysosome. These are circular structures, spherical sac. They basically carry a lot of hydrolytic enzymes. We discuss this later, each as when we're discussing the organelles. We have centrioles. Now, how the centrioles appear in the animal cell, the ultrastructure, we see there are two centrioles or a pair of centrioles, and the centrioles are there as a one cylinder and the other cylinder, both at right angles to one another. And the region where they are present, this is called as the centrosome. Around the centrosome, we have microtubules radiating all along the cell. In animal cells, we have microvilli as well, and these microvilli, they increase the surface area for absorption. So these are the extensions of the cell surface membrane. And inside the microvilli, we have microtubules as well to strengthen the microvilli. And the same thing goes for cilia and flagella as well, which we'll discuss later. So moving on to Golgi complex, we have Golgi body. Golgi body are these tubular structures. These are Golgi body, and the rounded ones are the Golgi vesicles. Together, we call these as Golgi complex or Golgi apparatus. You have cell surface membrane as well. So this pretty much covers up all the structures that you can generally see in an animal cell. This is the transmission electron microscope image of an animal cell. You can see the nucleus and you can see the mitochondria, the nuclear envelope, and the cell surface membrane here. So all these structures can be seen. And this is the transmission electron micrograph because internal details were basically visible here. Moving on to the first organelle in detail, we are discussing nucleus. Nucleus. What's the function of the nucleus? Your nucleus basically contain your DNA, and DNA holds the genetic blueprint and the hereditary information for an organism. And this information is passed on from one generation of the cell to the next generation of the cell during cell division. If you look at the nucleus, we see nuclear envelope, and the nuclear envelope can be seen very clearly. This structure here is the nucleolus. These structures here are the chromatin, and the gaps and the pores that you see along the sides of the nuclear envelope, these are nuclear pore. Nuclear pore allow exchange of materials between nucleus and the cytoplasm. As already I've discussed with you, nucleus is the site where the ribosomes are synthesized. So this is an example that ribosomes, once they are produced, they move out of the nucleus using the nuclear pore and they go to the cytoplasm. Outside the nucleus, you can see these are the rough endoplasmic reticulum, and you can see these very, very clearly here. This structure here, apparently, it seems to be like a lysosome. These both look like mitochondria because internally you can see compartments and these details are seen are visible here. So these should be mitochondria. Whereas this one, which I have marked it as an A, this appears to be like a lysosome because internal details are not there. There is not much contrast present inside this diagram. Chromatin, nucleus, nuclear envelope, nuclear pore. This is what we have discussed. So you need to understand the use of two terms, chromatin and chromosome. You can't use them interchangeably. Chromatin is composed. What is chromatin? Chromatin is composed of DNA and RNA as well as proteins, and they appear as granules or fibers or patches in an electron microscope. Chromosome, on the other hand, is tightly coiled chromatin and is visible only during cell division and in eukaryotes, as well as visible in bacteria. Nucleolus, already discussed with you, the function of nucleolus was to manufacture ribosome. It is having its own DNA, so yes, it can make its own ribosome. It basically make ribosome for the cells. What nucleus actually looks like? It looks like a densely stained small structure present inside the nucleus, composed of DNA, RNA, and protein. So these facts should be kept in mind. Moving on to the next organelle is rough endoplasmic reticulum. Wherever you have to mention rough endoplasmic reticulum, although while speaking, we basically end up using this term very often as rough ER or RER. Never use this in writing in your answer, or it will be rejected. So what you need to understand every time you use the name of this organelle, you have to write rough endoplasmic reticulum in full form. Rough endoplasmic reticulum is made up of cisternae, and cisternae are these tubular structures that are basically forming the rough endoplasmic reticulum. What are cisternae? Cisternae are membrane-bound flattened sacs, and they are fluid-filled cavities, and they are bounded by membranes. So this entire structure is a fluid-filled cavity. The line that I am just drawing here in red, this is the region of membrane of cisternae, and inside there is a fluid-filled cavity inside the cistern. So this is one cistern of your rough endoplasmic reticulum. This is the other and the other and the other and the other and so on and so forth. And this will be always connected with the nuclear envelope. In this case, this is the nuclear envelope. These are the chromatin you can see inside the nucleus. This structure here is what I've discussed with you earlier. This is centriole. One of the cylinder appears vertical, the other appears circular. So a pair of cylinders can be seen in this diagram as well. So coming back to rough ER, in rough ER we have the cisternae, and on the surface of cisternae you have ribosomes attached, and these ribosomes and the rough ER together they carry out the process of protein synthesis. The ribosomes that are attached here are called ATS ribosome. What is ATS? We'll come to this a bit later. Now, coming back to cisterns, the cisternae of rough ER are membrane-bound flattened sacs. These are fluid-filled cavity. They are responsible for carrying these proteins in the form of temporary vacuoles or vesicles to Golgi. Golgi is another organelle inside the cell, and the function of Golgi is to modify the products of ER. Anything that ER produces will be sent to Golgi, and Golgi will end up modifying it. So how this ER can send things to Golgi? So this the cisternae ends up being pinched off into tiny vesicles. The vesicles then start to move within the cell, and they go to Golgi and they fuse with Golgi. This is diagram showing to you how the rough and the smooth ER, they both are joined to the nuclear envelope, and they both can pinch off into tiny vesicles, and these vesicles will carry their products to the Golgi for modification. This is the TEM or rough ER. This is the TEM of smooth ER. The TEM of rough ER shows to you how the tubular nature of cisternae of the rough ER looks like, how they are having ribosomes on the surface as well. But when you discuss this smooth ER is very tricky for many students to identify on diagram. It appears, the cisterns appear more curved and more rounded, and you can say They are more bulgy at the end, and they do not have any ribosomes on their surface. So all these are your smooth ERs, all these rounded structures. These are all the cisternae of the smooth ER. You do not see these kind of rounded structures or cisternae in this rough ER. Although they both have cisternae, but the shape of the cisternae of rough ER and smooth ER is not exactly the same, although they both are made of cisternae. So the rough endoplasmic reticulum and smooth endoplasmic reticulum. This is the rough endoplasmic reticulum I have drawn. This is the smooth endoplasmic reticulum I have drawn. So what you see in a rough endoplasmic reticulum, the product, which is a protein, is being made. Once it is made, a tiny vesicle will pinch off and it will go to Golgi for modification. Similarly, the product produced in smooth ER, they get pinched off as a temporary vesicle, the vacuoles, to Golgi as well, where they basically undergo modification as well. Rough ER produce proteins, we know that. Smooth ER, the function of smooth ER is to produce chemicals that carry out detoxification, producing lipids such as steroids and cholesterols. They are all produced inside smooth ER. These are some examples that I mentioned where you can find a lot of smooth ER because it is involved in these processes where lipids are being synthesized and detoxifying chemicals are being produced. Moving on to ribosomes. Ribosomes are attached to the rough endoplasmic reticulum as we have just discussed. So ribosomes, what do we already know about ribosomes? Ribosomes are tiny organelles. They carry out protein synthesis, and they basically have two types. Prokaryotes have ribosomes which are called 70S ribosomes, whereas eukaryotes also have 70S ribosome together with 80S ribosome. So eukaryotes carry both ribosomes. 70S ribosome inside a eukaryotic cell is present inside their mitochondria and inside the chloroplast. Whereas the ATS ribosome in a eukaryotic cell is present in the cytoplasm, the rough endoplasmic reticulum, and the nuclear membrane. Summarizing this thing here, ribosome, two major types, 70S and ATS. S basically is the unit for size, and basically it's the unit for sedimentation. A heavier ribosome sediments faster in experiments. A lighter ribosome sediments slower in the experiment when we try to sediment these ribosomes in a centrifuge. So ATS ribosome is a heavier ribosome, a larger ribosome having a diameter of 25 nanometer. 70S ribosome is a much smaller ribosome having a diameter of 20 nanometer. Both ribosomes are composed of ribosomal RNA and protein. This is what they are made up of. They lack membrane. They are among the organelles who do not have any membrane. Every ribosome will have a large subunit and a small subunit, and together these large and small subunit will make up either the 70S ribosome or they will make up the ATS ribosome. So both the ribosomes have two subunits. This is what the ribosome is doing. This is the information that we discuss when we are discussing protein synthesis. For now, I'll skip this. Coming to Golgi. What is Golgi complex? Golgi complex is also made up of the same kind of cisternae that your rough and smooth ER is made up of. Golgi complex is having two basic structures, cisternae and the Golgi vesicles. All these structures are cisternae. And at the end, these cisternae are constantly budding off to form vesicles, and the ends of these cisternae are bulging and they are rounded and they are pinching out and they are forming temporary vacuoles, carrying the modified products of ER away from the Golgi. So this is what Golgi is doing. Golgi is also made of cisternae, and the cisternae of Golgi are saucer shaped. We use the term saucer shaped here, and these saucer shaped cisternae are stacked on one on top of the other, and this is what the Golgi complex looks like. When you study Golgi, these are stacks of membrane bound fluid filled flat sac, which is cisternae. Constantly, these are forming at one end, constantly, they are breaking off as vesicles at the other end. You can see this in the diagram. This is all Golgi complex. At the end, the Golgi complex are just separating into different vesicles. All vesicles are not of the same sizes. Some are large, some are small. And for example, this particular cistern last is actually completely disintegrated. So Golgi keeps forming, keeps maturing, and keeps breaking. So this is a continuous process. What is the function of Golgi? Let's just recall Golgi is visible under a high power light microscope. Yes, it is visible because it's larger than 200 nanometer, whereas rough and smooth ER are not visible. Ribosome is not visible in the light microscope. Nuclear pore is not visible. So you need to make a list of organelles and the structures which are visible in the light microscope and those which are visible in the electron microscope. Coming to Golgi, as continuing. So Golgi, what's the function of Golgi? The function of Golgi is to collect, modify, package, and sort. These four basic functions of Golgi are to carry out these processes for the ER products, which could either be from rough ER or from smooth ER. Apart from this, in plants, Golgi perform an additional function of producing those sugars which are cell wall components. This diagram here sums up how the proteins are made on the ribosome in the rough ER, how a vesicle will go and fuse with the Golgi, delivering the protein to the Golgi, how the Golgi will allow it to be modified. How can a protein be modified from Golgi? There are various ways in a protein which a protein can be modified. We know proteins are the chain of amino acids. So if a carbohydrate group is added to that protein, it means that the protein underwent some modification. So this process is an example of modification of protein inside Golgi. There are various other processes by which a protein can get modified in Golgi. You study those later on in the syllabus, but for now you can just remember that adding a carbohydrate chain to a growing chain of amino acids protein in the Golgi complex is an example of modification of protein. Now, the products of Golgi are all taken inside these vesicles. All vesicles carry different kind of products. So all vesicles will not release their products outside. Some of the vesicles will stay within the cytoplasm for the products to be used within the cell. So all the modified products of endoplasmic reticulum that are packaged in the Golgi vesicles, they can be used inside the cell as well as outside the cell. So this pretty much covers up what I wanted to discuss in this video. I'll be discussing remaining organelles in the next one. I hope this video was helpful for you. That's it from my side. Thank you so much.